My name is Curt Scharfe, and I'm a faculty member at the genetics department at Yale School of Medicine. The title of my talk is Reducing False Positive Results in Newborn Metabolic Screening Using Machine Learning. Financial disclosures, I have nothing to disclose. We have three learning objectives today. First, newborn screening, the principal approach. Second, the challenges of false positive screens. And third, introduction into novel data analysis and web-based tools. Newborn metabolic screening utilizes dried blood spot cards. They are collected 24 to 48 hours after birth. These cards are sent to state newborn screening labs for mass spectrometry-based screening of metabolic disorders. Results are either negative or positive, and positive reports are reported back to the hospitals to perform confirmatory testing. During confirmatory testing, some of these initially screened positives may turn out to be false positives, and they're excluded. Others are confirmed, and treatment is initiated for patient care and management. Results are reported back to the newborn screening programs. Here on the right side, you see an example of two blood spots from two babies and 3 millimeter punches made for analysis. You need to keep in mind that screening is not equal to diagnostics. That means we will have to have confirmation of all screen positives during the process. A major challenge in newborn screening are false positive screens. And here is an example given for four disorders, where you see the proportion between true positives and false positives. These data were collected over a period of 10 years in the California newborn screening program. The four disorders are glutaric acidemia type 1, methylmalonic acidemia MMA, and ornithine transcarbamylase deficiency OTC, and very long chain acyl coenzyme A dehydrogenase deficiency. As you can see, this large number of false positives for each disorder leads to a lower positive predictive value, which is the ratio of true positives over true positives plus false positives, ranging from 3% to 23%. Here's an overview over the recommended uniform screening panel, which contains currently 35 core and 26 secondary conditions. And the four disorders we have selected here are listed. Three of them are on the RUSP, they're metabolic conditions, and one disease is currently not on the RUSP, is OTCD, which is screened in California since 2010. Changes in metabolic analyte levels for selected metabolic disorders are shown here. So the MS panels contain some 40 metabolic analytes. Those include amino acids and isocarnitine analytes. For the four diseases shown here are the analytes which are used as primary screening markers. So for example, for methylmalonic acidemia, C3 and the ratio of C3 and C2 is being used, and if they're elevated, these babies are being flagged as screen positive. Metabolic analyte levels are influenced by several clinical variables or covariates. NBS programs collect these information about these variables at the birthing hospital. For example, there is birth weight recorded, gestational age, sex, race, ethnicity, age at blood collection, and total parenteral nutrition, which are filled out here on these cards with every newborn screen. This information may be later used in the interpretation of newborn screening data in the newborn screening reference laboratory. It may also be used to flag potential false positive cases, which could help in the interpretation, for example, of preterm babies or babies which receive total parenteral nutrition. Here's an example of the influence of birth weight on the y-axis and gestational age on the x-axis for metabolic marker levels. In this analysis, we studied a cohort of 100,000 screen negatives, healthy newborns, which we divided into 25 groups defined by birth weight and by gestational age. The color code indicates the median levels of a particular marker. In this case, it is propionyl carnitine C3 for each of the groups, with the group size shown in parentheses. As you can see, the levels of these markers are quite different between these different groups. So there is a confounding effect of marker levels independence of birth weight and gestational age. Plots for other newborn screening metabolic analytes are available at the web address listed below. Finding links between metabolic markers and the various clinical variables and disease risk is very difficult for a person, but perhaps well suited for machine learning algorithm. Machine learning is an emerging strategy for classification of metabolic disorders. Here's a simple introduction to machine learning concepts. There are, in principle, two main types of machine learning: supervised and unsupervised learning. So what's the difference? Supervised learning has a ground truth. There is a prior knowledge of the input. For example, there is a label for false positives or true positive cases, which we can use for training and testing in the classification of data. In contrast, unsupervised learning has no labels and there is no training. We can infer the structure in the data set to the data itself to provide initial insights. A commonly used unsupervised method is clustering. In this cluster analysis, for example, the two ducks cluster together. And this could be a different cluster analysis of the same data. Here, the non-carrot eaters cluster together, separate from the carrot eater. Unsupervised learning is entirely based on the data. We don't know which samples will be clustered together before the clustering. And we may have different clusters based on different criteria depending on the methods being used. Here's an example, an unpublished example from our work for using unsupervised learning for age and blood collection data. In this analysis, we studied NBS data from roughly 500,000 babies for 41 metabolites shown on the y-axis and across different age at collection time points ranging from 12 till 72 hours after birth, shown on the x-axis. The differences in metabolite levels between the different time points were measured using effect size analysis. The measure we are using is Cohen's d. The first time point, 12 till 17 hours, not shown here, was used as a standard. Looking at this clustering figure, we can identify two large clusters. In blue on the top, there is a decrease in metabolite levels over the time course. Examples for metabolites following that trend are phenylalanine and methionine. So they will decrease over the course after birth. On the bottom in red, there is an increase in levels. Examples are valine and leucine, isoleucine. So these metabolites will increase after birth. No group labels were used in this analysis, so we are not using any information here in addition to the markers. Only the metabolite data are being used. We don't have a ground truth or a reference as to which metabolites would group together prior to doing this clustering. Thus, it is a largely exploratory, unsupervised approach. However, it may provide some insights that could be used to test individual hypotheses. Now, in comparison, supervised learning is typically done with data labels. The goal is to find specific relationships or structure in the input data that allow us to effectively produce correct output data. On the right, for example, we can perform a classification of categorical labels like false positives or true positives for a specific newborn screening disease to separate these two groups from each other. Typically, we need to train the model in order to get the best performance for separating these two groups. In this example, we may get the dashed lines at first analysis. We then compare the results to the labels, which is the truths, to check the performance of our new model, and then accordingly adjust the model to find the best fit, which is shown as the solid line. However, one challenge in supervised learning is overfitting, shown in this not straight line shown here in the model. Thus, we need testing data that are completely separate from training data. However, newborn screening disorders are typically rare, and sometimes we may not have enough data available for the training set data. In such cases, cross-validation is being used as an alternative way to estimate the performance of the model. Cross-validation is used to estimate the performance of the supervised learning model. In principle, using cross-validation, we can separate the data into different sets, each containing, for example, a number of true positive and false positive cases. In this example, a relative larger number of false positives versus true positives in each group. We divided, in this example, the data into 5 groups of 5 false positive and true positives. At each cross-validation step, 4 of the 5 groups are combined for training, shown in gray, while only 1 group is used for testing, shown in yellow. After the 5-fold cross-validation, each of the 5 groups has been tested. Predictions, importantly, are only made for the testing set, not for the training set. Random forest or random decision forests are very powerful tree-based methods for supervised machine learning that have been developed for high-throughput genomic and metabolomic data analysis. We have recently shown an analysis of tandem mass-spectrometry data obtained from the California Newborn Screening Program using RF, which was able to improve the separation of true and false positive cases for one disease, methylmalonic acidemia. In a recent analysis published in 2020, we applied a random forest to the study of additional metabolic disorders to improve the diagnostic of glutaric acidemia type 1, VLCAT, and to facilitate the detection of OTC, which is, as I mentioned, not currently on the RUSP. The performance and the stability of our RF model was evaluated using newborn screening data from screen-positive infants for these 4 disorders reported by the California Newborn Screening Program. To estimate the reliability of RF to correctly predict true and false positive cases, we used a leave one out cross-validation approach. For example, for each disease, the RF model was trained on all screen-positive cases for that disease except for one blinded case for which we made a prediction. This process is then repeated for all screen positives for that particular disease. As an example, 604 of the 605 screen positive MMA cases were combined for training, while only 1 MMA screen positive case was blinded and used for testing. This process was then repeated until all 605 cases were classified by RF. These figures show the results from our analysis of newborn metabolic profiles for the 4 diseases with random forest. The receiver operating characteristics curve analysis was plotted for all infants with and without a confirmed diagnosis using RF analysis of 40 MS analytes. Without altering the sensitivity of primary newborn screening, for each of the 4 disorders, RF was able to reduce the number of false positive cases, and that is the vertical dotted line in red, by 89% for GA1 45% for MMA, 98% for OTCD, and by 2% for VLCAT. For each disease, the number in parentheses shows the 95% confidence interval of the area under the ROC curve. Random forest is a bit of a black box. However, there are ways to measure the contribution of individual metabolic markers or analytes made by RF in the predictions. Here we used the mean degrees in accuracy, MDA, to rank the relative importance of individual mass spectrometry analytes in clinical variables for metabolic pattern recognition in our RF model. In this figure, only the 20 top-ranked analytes and variables are shown for each disease. The primary screening marker for each disease are labeled in red. For example, C2 and C3 for MMA and the panel B. In theory, this MDA analysis could also be used as a tool for biomarker discovery by looking at the top-ranked markers, which are not yet being used as screening markers. This table is taken from our paper and shows the 5 top-ranked analytes identified by a random forest for each disease. Notably, each of the primary markers used to detect these 4 diseases in the California newborn screening program was among the 5 top-ranked analytes for each disorder. 2 of the top-ranked analytes are part of informative ratios for GA1, C8, and MMA methionine, while several other analytes were found to be related to these disorders based on literature searches. A novel web-based tool using a Shiny app was developed in this project that now enables interpretive analysis of MS/MS data from newborn screening using random forest. Here shown is the graphical user interface of this new web-based RF tool for NBS data analysis. The menu panel on the left side was designed to upload data and to select parameters. The panel on the right side shows the result from RF-based analysis of the metabolic input data, metabolic user input data. Users can select a specific cutoff value or use the default cutoff, which we calculated for each disorder based on the median sensitivity of 1,000 repeats in a cross-validation, in a tenfold cross-validation. For example, moving the slider to the left in the left frame would reduce overall screening sensitivity, more false negatives, and reduce also the number of false positives in the test. The web address for this new tool is shown below. In summary, there's limitations of our approaches, and I like to line out a few future plans. Our RF tools database currently has only a very limited number of samples, less than 3,000 samples of true positive and false positive for 4 diseases, and only from 1 newborn screening program. So we are interested to expand this database with new sample data from diverse patient populations. And second, the number of metabolites is currently 41 per sample, so 41 metabolites measured at birth in each baby for metabolic screening. It is still limited because it's based on, it is utilizing targeted MS/MS data. We're interested to develop untargeted metabolomics method to get a more fine-grained picture or metabolic profile of the newborn. We are also working on integrating metabolomics data, targeted and untargeted metabolomics data from blood spots with genomic sequencing data and other omics data. This project has been a collaboration between investigators at Yale and at Stanford and the California Newborn Screening Program. Funding was received from the National Institute of Child Health and Human Development. Please contact me if you have any questions or suggestions. Thank you for your attention.